Hello everyone, my name is Anthony. Today for my Orison presentations, part 3 of exam 2, I would like to address two questions. Question 1, explain how the mechanism of fungal toxin peripherin A affect the retrieval of KDEL tag protein from cis Golgi network back to the ER. First, the definition of endoplasmic reticulum is consisted of cisternae and space enclosed by the ER membrane called the lumen. They have two types of ER. Rough ER is they have ribosome synthesize polypeptide to protein within the membrane, then transport to the ER lumen. This protein travel to the membrane of the cell surface by using secretory vesicle from Golgi apparatus. Smooth ER by the name has no ribosome, no role in protein synthesis, but they synthesize steroid, detoxify drugs, carbohydrate metabolism, and calcium storage. This is a picture of what the rough ER look like. On the left hand side, you can see that rough ER on the top and the smooth ER in the bottom. And then on the right hand side, we have many blue dots in between of the rough ER. Those are free vesicle contain of protein and lipids ready to be delivered to the Golgi apparatus. Golgi apparatus consists of stack of cisternae, which is flattened sacs, similar to the ER. There are two phases of the cisternae. First, the cis Golgi network, which consists of a network tubule that located close to the ER. Vesicle inside the CGN network contain a protein and lipids that arrive from the ER. And for the trans Golgi network, also consists of the tubule, and the function have to transport vesicle that contain of lipid and protein to endosome, lysosome, and plasma membrane. This is a picture of what the Golgi apparatus look like. On top, we have the cis Golgi network with vesicle arrive from the ER, and on the bottom we have the trans Golgi network with the vesicle that we ready to be transported out to the other membrane bound like lysosome, endosome, or even to outside the cell by exocytosis. I want to talk a little bit about anti-Guray and retrograde transport. Anti-Guray transport is when the material from the ER move to the Golgi and toward the plasma membrane. But in opposite, retrograde movement is the movement of vesicle from the Golgi apparatus back to the ER. So that promotes the ER retention. This picture is how the protein traffic inside the endomembranous system look like. We saw on the left side we have the ER synthesize protein and lipids and deliver it to vesicle and deliver it to the Golgi apparatus and eventually to the surface of the plasma membrane for exocytosis to outside the cell. That is considered anterograde transport. In the bottom, we have the retrograde transport, which endocytosis of the cell surface, moving the vesicle back inside the endomembranous system to first to early endosome, to late endosome, and to lysosome. So how ER have retrieval tag promote the ER retention of that protein? First, each protein in the ER contains a specific tag that target the protein to the vesicle before they make it carry it from one specific cellular location to another. These tag helps to retain ER because they retrieve protein that have left the ER and move into the Golgi apparatus. So pretty much the vesicle that contain protein moving back from the Golgi apparatus, the cis Golgi network, back to the ER. The sample of those tags, one of the samples is called the tripeptide sequence RXR, which stands for arginine X arginine amino acids. X is skipping any amino acids. Another tag protein is called KDEL. This is the full name is lysine, aspartic acid, glutamic acid, and leucine. This tag protein promotes retention in ER mammals in comparison to different kind of organisms. They have the short C-terminal amino acid sequence. They bind to transmembrane receptor facing the Golgi apparatus and inducing the conformational change and cause the receptor-like complex that fully packed with vesicle of protein return back to the ER. So that's considered a retrograde movement right there. Another protein called disulfide isomerase and BIP to ER. These proteins help to process the KDEL sequence that promote retention in the ER. So does Golgi have the same function? Yes, they do. they contain retention and retrieval tag for their resident protein as well. They call hydrophobic membrane spanning domain. These protein help to move they move from the cis to the trans Golgi network until they observe the thickness of the membrane that go increase and beyond the length of the hydrophobic membrane spanning domain, and therefore they block any further migration or that would be able to retrieve their resident protein. So COPI, coated vesicle, what is that? They're found in eukaryote cells, and it will really support the retrograde transporter protein from the Golgi apparatus back to the ER, as I described earlier, and it also support the bidirectional transport between Golgi apparatus cysteinate. They compose of COPI protein, COP1 protein of 7 subunits, and the EDP stands for ribosylation factors occurs as part of the ARF GDP complex. So this ARF is also considered a GDP binding protein because they encounter guanine nucleotide factor in the membrane of new coated vesicle and help that to exchange GDP for GTP. That actually induce a conformational change of ARF, and when exposed the hydrophobic N-terminal, which attach to the lipid barrier of the membrane. And this ARF factor really mediate the COP1 protein because when it binds to the COP1 protein, it starts the formation and budding of new vesicle. This protein will trigger the hydrolysis of GTP and transform it back to the ARF GDP complex as originally and release the co-protein, repeat the cycle again. So how fungal toxin Prefidin A inhibit that process? First, Prefidin A is a lactose antiviral produced by penicillin perfidinium. They inhibit the retrieval of KDL tag protein from the cis Golgi network back to the ER by prevent the association of COP1 code to the Golgi membrane. So how is it really working specifically? Well, the fungal toxin, the Prefidin A, inhibit the guanine nucleotide factor to produce ARF-GTP from ARF-GTP, as I described earlier. It blocks the process, so the vesicle from the Golgi apparatus can no longer be delivered back to the ER by the retrograde movement anymore. So that's how it works. Question 2, describe how low concentration of Botox can be used as a treatment for wrinkles and muscle spasm. Well, first, what does it mean by the Clostridium botulinum bacteria? It can be found anywhere, such as soil, fruit, vegetable, and the air. They can grow in anaerobic environment to produce the botulinum toxin. There are 7 main types of this kind of toxin, and the botulism is the disease when people ingest toxin that they eat from food that's not well preserved. Botox, what is that? It's the one of the 7 main type of the toxin. This is type A. They are paralytic toxin produced by the bacteria Clostridium botulinum, as I described earlier. They help to enhance youthful appearances, especially people who are not trying to go through plastic surgery and they're trying to reduce aging, they're trying to look young. So this is the structure of how the toxin A look like. We saw the translocation in their domain, receptor binding domain, heavy light change, and cleavage size. So pretty much that the translocation domain uptake in the cell through endocytosis, right? And the toxin presented here is in inactive form. So when it's going through the reducing environment of the endosome, it will cleave the disulfide bond and it will release the catalytic light chain into the cell. Well, to make better sense of it, let's take a look at these pictures. This is how the toxin A attack to the neuron cell versus the picture on the left side is how the normal mechanism of transmitter release inside a neuron look like. We can see right there, we have the synaptic vesicle connected to many different kind of protein. We have SNAP protein, we have SNAP25, and eventually those vesicles will be containing acetylcholine will come to contact with the cell sulfate of the plasma membrane and release the acetylcholine and induce muscle contraction of the muscle cell. But once the toxin A in the light chain of the toxin A enter the neuron cell, we will see that they will cleave the protein. So those those protein, those snail protein, the SNAP25, it can no longer bind to the cell surface anymore. So instead, the vesicle would be blocked, and the acetylcholine can no longer be delivered to the muscle cell, and that would block the muscle contraction, which is really the deadly conditions. But in treatment, how is that Botox for treatment for wrinkle and muscle spasm? Well, we can use a small concentration of Botox to induce local paralysis of specific region of the face. The wrinkle can diminish because the muscle can no longer contract under the skin, as I described earlier, due to that little guy is the choline can no longer bind to the cell surface, so the muscle cell can no longer be contracting. The benefit of this Botox treatment is that we will get the better result of the skin, smoother, less wrinkles. But the downside is that it's not going to be long lasting, only the few weeks to a few months. That should be about it. Thank you. This is my reference.